Assalamu alaikum dear students. Welcome to the course Principle in Animal Life 2. This is lecture number 10. The topic is the Law of Independent Assortment. Discussing agenda for the topic will be the Law of Independent Assortment. Lecture outcome: after watching and listening this lecture, students will know about Mendel's Law of Independent Assortment and examples. Introduction. Mendel derived the law of segregation from experiments in which he followed only a single character, such as flower color. Mendel's first law states that each trait is determined by a pair of factors. The factors stay together in a diploid individual but are separated or segregated during gamete formation and are reunited at the time of fertilization. So, in order to arrive at the conclusion, Mendel first studied only a single character. He crossed parental varieties, true breeding. They were in nature and were differing only in a single character like seed color, seed shape, flower color, height, flower position. So only one trait was under consideration in those crosses, and those crosses are also known as monohybrid crosses because the parental varieties were differing only in a single character. All the F1 progeny, the F1 hybrids that were produced as a result of these crosses of true breeding parents were monohybrids, that is, they were differing only in one particular character which was under observation in the cross. We refer to cross between such heterozygotes which are differing only in a single character as monohybrid cross. Mendel worked out the second law of inheritance by following two characters at the same time, such as seed color and seed shape. So the law of independent assortment, where two characters were studied at a time, and Mendel performed dihybrid crosses. That is, the parental varieties were true breeding, but they were differing in two characters. Now, seeds may be either yellow or green. They may be round or wrinkled. So these two characters were under consideration, and thereafter Mendel deduced his findings and formulated them into a law which is known as the Law of Independent Assortment. From single character crosses, Mendel knew that allele for yellow seed is dominant and the allele for green seed is recessive. So during the experiments that formally resulted in the development of the first law, Mendel crossed plants that were differing in various characters, but he studied each character at a time, only a single character at a time. So from the experiments, earlier experiments, Mendel already knew which character is dominant and which one is recessive. So yellow seed color was dominant over green. Similarly, the round seed shape was dominant over the wrinkled seed shape. Now imagine crossing two true breeding. True breeding means the parental varieties which are homozygous for all the traits. So the cross between true breeding varieties that differ in two of these characters, that is the seed color and the seed shape. So we write it as the genotype for yellow round seeds will be this one, and the genotype for the recessive homozygous recessive, that is green wrinkled seed, will be this one. So now what will happen? What will be the progeny of the F1? What will be the genotype of the F1 hybrids? And what will happen when the F1 hybrids are crossed together? They are selfed together. The F1 plants will be dihybrid, differing in two characters. So this is the genotype of the F1 hybrids. Now the phenotype will express the phenotype of the dominant parent. So these F1 hybrids are having yellow seeds and round in shape, which is a dominant character. But are these two characters transmitted from parents to offsprings as package? So the next thing Mendel wanted to know was that whether the dominant allele for one character will always stay with the dominant allele for another character, and the recessive allele for one character will always stay together with the recessive allele of another character. So whether the dominant are transmitted together, or the dominant allele of one character can become part of a gamete which carries the recessive allele for another character. So he basically wanted to know that whether the grouping of these alleles in the gametes is independent or dependent. That is, well, the Y and R, which are the dominant alleles, they will stay together generation after generation. Similarly, the small y and small r, which are the recessive alleles, will they stay together generation after generation? Or are seed color and seed shape inherited independently? So whether the alleles are independent, they are not linked with one another. So obviously later on researchers studied gene linkage, which violates the second law of Mendel. Gene linkage states that the genes located closer together on a chromosome will be inherited together. But here the Mendel law experimentally proved that the alleles controlling different characters are segregated and they form groups independently of each other. Means it is not necessary that a dominant allele of one character will always be together with a dominant allele of another character. Similarly, it is not necessary that a recessive allele of one character will always be together with the recessive allele of another character in gametes. The grouping is independent. Assortment means grouping, so this grouping is independent, means the dominant allele of one character can become part of a gamete which carries the dominant allele of another character or the recessive allele of another character. So this grouping is independent. There is freedom. In the last two slides we will discuss this whole cross diagrammatically. So a cross between F1 dihybrids can determine which of these two hypotheses is correct, means whether the alleles determining different traits are independent or they are dependent. They are closely linked with each other. They are inherited together. So the F1 plants of genotype heterozygous dominant for color which is yellow and heterozygous dominant for seed shape which is round exhibit both dominant phenotype yellow seeds with round shape, no matter which hypothesis is correct. So the key step in the experiment is to see what happens when F1 plants self-pollinating and produce F2 individuals. Now the F1 hybrids will always carry this genotype because there must be contribution from the recessive parent. Now the point will be cleared when the F1 hybrids are crossed with each other, and thereafter it will become clear whether the dominant allele of one character has inherited together with the dominant allele of another character or the grouping was independent. If the hybrids must transmit their alleles in the same combination in which the alleles were inherited from the P generation, that is parents, then the F1 hybrids will produce only two classes of gametes. So if we predict that the dominant allele of one character always stay together with the dominant allele of another character, and the recessive allele of one character always stay together with the recessive allele of another character, then only these two types of gametes are possible, both sperm and eggs. Now, independent assortment hypothesis predicts that the phenotypic ratio of the F2 generation will be 3:1, just as in a monohybrid cross. That is, three parts will be having yellow color and round seed shape, whereas one part will be having green color and wrinkle seed shape. So we will explore in the next few slides what will be the outcome of the F2 in a dihybrid cross. The alternative hypothesis is that the two pairs of alleles segregate or group together independently of each other. That is, a dominant allele of one character can become part of a gamete which carries the recessive allele for another character. In other words, genes are packaged into gametes in all possible allelic combinations. So the grouping is independent as long as each gamete has one allele for each gene. Now in our example, on if one plant will produce 4 classes of gametes, 4 classes of gametes will be produced only when there is no gene linkage. When there is independent grouping, you can see here all types of allele combination is possible. Dominant for one character along with dominant for another, dominant for one character along with recessive of another. Similarly, the case is here, so all types of allele combination is possible, and that only happens when there is no linkage between the genes. And luckily for Mendel, there was no linkage observed in these 2 characters, otherwise it would have been problematic for him to explain. Later on, researchers, particularly T.H. Morgan, studied gene linkage and it proved to be an exception to Mendelism or Mendelian genetics. If sperm of the 4 classes fertilize eggs of the 4 classes, there will be 16 equal probable ways in which the alleles can combine in the F2 generation. Now you can construct a Punnett square so Y or Y or Y or and Y or. Similarly, you should write the gametes here. Now you can make a simple Punnett square and then you will find out the progeny. You can construct a Punnett square These combinations result in 4 phenotypic categories with a ratio of 9:3:3:3:1. That is 9 yellow round, 3 green round, 3 yellow wrinkled and 1 green wrinkled. When Mendel did the experiment and classified the two offsprings, his results were closer to the predicted 9:3:3:1 phenotypic ratio, supporting the hypothesis that the alleles for one gene controlling seed color, for example, segregate into gametes independently of the alleles of any other gene, such as seed shape. So the grouping of alleles in the gametes is independent only when there is no linkage, which was the case in Mendel's experiments. Mendel tested his 7 P characters in various dihybrid combination and always got this phenotypic ratio in F2 generation. So it means for all the characters Mendel studied, there was no linkage observed. Now is the 3:1, which was the outcome of the F2 generation in first law. So how this 9:3:3:1 relates with 3:1, you can calculate it by observing the yellow and green seeds, ignoring the seed shape, so you will get the same ratio. Now the result of Mendel's dihybrid experiments are the basis for what we now call the law of independent assortment. How we state this law: two or more genes assort independently, group independently into the gametes. That is, each pair of alleles segregate independently of any other pair of alleles during gamete formation. So the grouping of alleles determining different characters is independent. This law applies only to genes located on different chromosomes, or alternatively to genes that are far apart on the same chromosome. Means if there is no linkage between the genes controlling different characters, only then they will segregate independently. If the genes controlling different characters are located closer to each other on a chromosome, then a crossing over event cannot separate them, and they will be grouped together in the same gamete. So this phenomena is called linkage, which is fortunately not observed in Mendel's pea plants. All the P characters Mendel chose for analysis were controlled by genes on different chromosomes or were far apart on the same chromosome. This situation greatly simplified interpretation of his multi-character P crosses. So fortunately for Mendel, he didn't came across a situation where there was gene linkage involved, therefore the interpretation of the data was much easier for him. All the examples we consider in the rest of this chapter involves gene located on different chromosomes. So now this is the pictorial representation of the law of independent assortment. So these are the two parental varieties, true breeding varieties, and this is a dihybrid cross. Two contrasting characters are under consideration. The yellow round is a dominant character, the green wrinkled is a recessive character. So this will be the gametes formed by the dominant parent, this will be the gametes formed by the recessive parent. Fertilization of these gametes will leads to heterozygous dominant. Now there are two hypotheses. Hypothesis is a tentative explanation to a question. The two hypotheses is here. Now if the gametes stay together, means the dominant allele of, if the alleles stay together, means the dominant allele of one character will always be together with the dominant allele of another character, and the recessive allele of one character will always be together with the recessive allele of another character, then we only get this. This will be dependent assortment. Dependent means dominant of one is dependent on dominant of another, recessive of one is dependent on recessive of another. So If the grouping is dependent, then we will get only two types of gametes. So then you can construct a Punnett square. On one side you should mention sperms, other side you should mention eggs. Then you construct a simple Punnett square and you will get this combination. So this is the phenotypic ratio. Now, if the grouping is independent, means it is not necessary that the F1 hybrids will produce gametes where the dominant allele for one character will always be together with the dominant allele of another character. Similarly, for the recessive alleles, the grouping is independent, so you get all kinds of allele combinations. You can see both dominant, one dominant, another recessive. Similar is the case here. Both are recessive. Sperms, 4 different combination of alleles. Eggs, 4 different combination of alleles. And when you construct a Punnett square and finally you deduce the results, you will get 9 yellow round, 3 green round, 3 yellow wrinkled, 1 green wrinkled. So all kinds of combination will occur. There will be different phenotypes. Some phenotypes will resemble the P generation parents, like the green round, yellow round, and green wrinkled. However, these two will be new allele combinations or non-parental varieties, and the new allele combination only arises when there is independent assortment. This is another simplified diagram where you can watch all the events happening. I hope you have understood the lecture. Thank you very much for your time. Allah Hafiz.